speaker is Lani Gleason. Introduce you. She's gonna talk about tabula on background of neutral similarity and in the marine name. Tabula for the back.
About my work using next-gen sequencing technology to identify genomic evidence for ecological divergence in the marine black turban snail, Potassium. Previous work has shown that many intertidal invertebrates, such as these petrolithes crabs, Vanillus mussels, Nucella whelks, Lani limpets, and the black turban snail, are living near the upper edge of their thermal limits, and so these species are at a particular risk for local extinctions, as anthropogenic climate change is likely to lead to even greater fluctuations in a variety of environmental parameters, including temperature. Because of this, we want to be able to predict the future population dynamics of these organisms in the context of global warming, but in order to do that, we need to know if all populations of a single species are going to respond in the same way. In other words, we need a better understanding of the balance between local adaptation, or the fine-tuning of individuals to their local habitat via natural selection, and gene flow, or the movement of alleles between populations via migration or dispersal. And in many marine invertebrates with planktonic larvae and fluctuating population sizes, the potential for local adaptation is unclear because we don't have this good understanding of the balance between the selection for local adaptation and the rate of interpopulation gene flow. As part of my dissertation, I've chosen to address this issue in one such marine invertebrate with planktonic larvae, the black turban snail, shown here, Potassium Channels. For my dissertation, I focused on individuals collected from 6 different sites in California, 3 in Northern California, Slide Ranch, Pescadero, and Pigeon Point, and 3 in Southern California, Aliso Beach, La Jolla, and Bird Rock. And in terms of geographic distance, these Northern and Southern California sites are separated by about 700 kilometers. And just to note, this is a color scheme I'll be using throughout the talk of blue to denote northern populations and red for southern. So one of the reasons we chose to do this work on the black turban snail is it's a really good system to address these factors that both promote and constrain local adaptation, because it's influenced by several different processes that contribute to both sides of this balance. For instance, on the one hand, we know it has a really wide geographic range, ranging from British Columbia in the north down to Baja California in the south. In addition, across this range, populations see wide variations in temperature. So here we're looking at a temperature record of estimated in situ body temperatures during the late summer and early fall of last year, which is typically when maximum annual temperatures are reached. And these are for the 6 different sites in California I just mentioned, and we can see the southern sites shown in red reach much higher temperatures and do so more frequently, including surpassing temperatures that we have shown in the lab cause 100% mortality in individuals collected from all 6 of these different sites. In contrast, at least during the time period of the study, the northern populations in blue don't reach this threshold. So we expect these 2 factors of the wide geographic range and the persistent temperature variation to promote local adaptation. And in fact, we have several different lines of evidence that indicate these populations are indeed locally adapted to heat stress. One of the first lines of evidence we have is from phenotype assays. So here we're looking at percent survival across a range of different heat stress temperatures. And we can see the southern population shows significantly higher survival. And just to point out here, this is that 41 degrees C threshold I showed in the last slide, where we see 100% mortality for all individuals. We also know that populations have unique patterns of gene expression under both control and heat stress conditions. So here we're looking at a PCA based on the transcriptome-wide gene expression of these 6 different populations. And we can see there are distinct and consistent differences between the 2 geographic regions. However, we also have evidence that indicates there could be gene flow occurring between populations which would preclude local adaptation. So previous work looking at the mitochondrial marker cytochrome oxidase subunit 1, or CO1, found no evidence for population structure in animals sampled from Oregon down to Santa Barbara. In addition, as I also briefly mentioned, we know this species has a pelagic larval phase with a duration of about 5 to 13 days. And these pelagic larvae could potentially facilitate dispersal and again work against local adaptation. So keeping these factors in mind, some of the research questions we wanted to address to gain a better understanding of the dynamics underlying this local adaptation. Well, first of all, do northern and southern California populations show evidence for genome-wide population structure? So we know there's no evidence at that single CO1 locus, but what if we look across the entire genome? And these genome-wide patterns will allow us to identify any potential homogenizing processes that could work against local adaptation. So for instance, if we don't see any evidence for population structure, this could indicate there are things like gene flow going on. And then to address the factors that promote local adaptation, we also wanted to see if we could find any genetic nucleotide level signatures of divergent selection, and if so, at what genes. And we're particularly interested in seeing if there are signatures at genes involved in response to heat stress, given our previous transcriptome and phenotype work. So we collected DNA from 15 individuals from each of those 6 populations, and then followed standard library protocols for DDRAD, or double digest restriction associated DNA, to Illumina sequencing, and then followed the STAX pipeline for SNP identification and population differentiation analysis. And for this analysis, we focused on regional differences. So we compared the 3 northern populations as a group to the 3 southern populations as a group, again based on our previous work that indicated this is the spatial scale we might expect to find differences. We then did subsequent analyses to look for population structure and identify potential outliers under divergent selection, and then use BLAST to annotate genes of interest. In terms of the data that we got back and the SNPs that we identified, we got back about 4 million reads per individual, which led to us surveying variation across about 2 million base pairs of the genome for each individual. Pooling by region resulted in the identification of about 394,000 total SNPs. And after assembly and quality filtering, we were left with about 2,300 SNPs. And then for the population genetics analysis, we did further filtering to retain a single SNP per locus, so we looked at about 1,600 SNPs for that. While for the outlier detection, we wanted to make sure we didn't miss any potential outliers, so we reran the data with different SNP subsets from the same loci, and ended up looking at about 1,800 SNPs for that analysis. So to give you an idea of the overall level of differentiation that we identified, here is an FST frequency distribution for the 2,300 high-quality filtered SNPs. These SNPs have an average FST about 0.0042, and for the distribution, there's a sharp peak at 0, and a long tail extending out to about 0.59. So for the first research question, looking for genome-wide evidence of population structure, one of the first things we did was a cluster analysis, and this is a structure plot showing the results of that analysis, with northern populations on the left, southern on the right. And in these plots, each column is a different individual, and the height of the colored bar represents the probability of an individual being assigned to one of the two clusters, in this case a red cluster or a blue cluster. And what we see here is a lack of a pattern. So for instance, we don't see all of the southern individuals assigned to one cluster, all the northern individuals assigned to another cluster. And if we look at this data in a slightly different way, this is a maximum likelihood snip tree where each branch shows an individual, southern individuals in red, northern in blue. Again, we do not see the geographic regions clustering into clades, but instead we see individuals from both regions spread pretty evenly throughout the tree. But we also did a separate analysis looking at different SNP subsets to get an idea of what proportion of the genome might be differentiated. So here we're just looking at the top 10 percentile of SNPs based on FST estimates across the 6 populations and seeing how they cluster into the 2 geographic regions. And now we're looking at 164 SNPs, and for the structure plots, now we do see evidence for geographic differentiation, where about 60% of the southern individuals are assigned to the red cluster, 70% of the northern individuals assigned to the blue cluster. And similarly for the SNP trees, now we see clades that are composed almost exclusively of southern individuals and vice versa. So next we wanted to see what those genes in that top 10 percentile were. So we were able to annotate 27 of them, which are shown here. Included in these genes are 3 ubiquitin protein ligases, and ubiquitins are involved in degrading proteins that are irreversibly damaged following heat stress. And it was interesting to find these genes because we also have evidence from our transcriptome data that indicates that the differential gene expression of these ubiquitins could be playing a role in the varying thermal tolerance of northern and southern California populations. In addition to just looking at that top 10 percentile of FSTs, we also used the program Locitan to identify potential outliers under divergent selection. Here we're looking at a plot of heterozygosity versus FST. Each of these different blue dots is a different SNP locus, and the shaded background colors on the graph are based on simulations run in Locitan. And I'll focus on this red region here, which are all potential outliers for positive selection. We identified 34 outliers and annotated 7 of them. In terms of their function, 2 are associated with potassium channels, the regulation of which has been proposed as an early event in initiation of the heat stress response. Another 3 of these are either known or hypothesized to be involved in stress granule formation. So a little bit more about stress granules and why they're important. What happens is when a cell is exposed to stress, such as heat stress, the housekeeping mRNAs exit the translating pool, and facilitated by microtubule transport and molecular motors such as dynin, they enter these stress granules, which are cytosolic aggregations of RNA and RNA-binding proteins that function to sequester and preserve these untranslated control mRNAs as part of a mechanism to facilitate the heat stress response. For instance, by ensuring the translation of other mRNAs like chaperones and enzymes that are immediately needed for the stress response. These stress granules also prevent apoptosis by sequestering pro-apoptosis factors and silencing their activities. What we think is going on here is that given their multitude of cellular stress survival functions, the formation of these stress granules is under selection in northern and southern California populations due to differences in thermal stress environment that they encounter. So to sum up, for the first research question, and when we consider all SNPs, we found no evidence for differentiation. But in a separate analysis, if we look at just the top 10 percentile SNPs, we do see geographic differentiation. And this includes genes involved in the ubiquity pathway. The second research question, looking for genetic evidence for local adaptation, we identified 34 outliers potentially under divergent selection, and 3 of those 7 annotated outliers are involved in stress gradient formation. So all this data together indicates that northern and southern California populations are undergoing the early stages of adaptive divergence, as evidenced by this genome-wide population homogeneity punctuated by strong genetic differentiation at several potentially ecologically relevant loci. Given the function of the genes that we identified as potentially under selection, we think that variation in thermal stress could be driving these differences. And lastly, more generally, these results also provide insight into the factors that promote and constrain local adaptation, especially in a marine invertebrate like this with pelagic larvae and no previously identified population structure. So with that, I'd like to acknowledge my lab and my funding, and if I have time,